What's up, guys? Lab 6. I'll tell you what we're going to go over. Special senses. We're going to talk about the eye, the ear. We're going to do a little histology. We're going to do some anatomy, learn the parts, a couple diseases, and a few tests for special senses that support what we learned or will learn in lecture. I gave you a list. Um, of some of the words that we're going to be talking about. We'll start with the eye anatomy. Most of the stuff probably already discussed. Um, Make sure you know everything on the list. We're going to start off. Obviously, I'll show you some models. Um, and kind of like this picture here, kind of shows it from the lateral view. I do have some of the models that I like, and I'll go through and I'll show you exactly where the structures are. Um, but clearly, we'll start from the front. And we will kind of don't like that part there, but it's all good. Um, as if a photon of light was passing in through the front of your eye, what structures it would hit, in what order, and a brief kind of explanation of what they are. So get rid of that guy. Light's coming in from this direction. The first structure we're going to hit will be the cornea. The cornea is clear. I'll show you a picture of it on the model. It is very important for starting to focus the light. We have some issues where you can see called an astigmatism, where that cornea is misshapen, or the eyeball in general, and you have a problem focusing light. As we work our way back, you will see, it's not the best on here, um, a space. This space is called the anterior chamber. I'll show you on a different picture. I don't know why it doesn't have it labeled there. But the anterior chamber is filled with fluid. The fluid is called aqueous humor. So we'll just make a note of that. It's called the anterior chamber, the anterior cavity. As this light moves through, it's going to pass through a space called the pupil. The pupil is just an opening. The opening is created by the iris. And the iris on here, you could see, change my color, pretty bad color. On this model is blue, and it's made of muscles. So these muscles can, and I'll show you at the end, constrict and dilate in response to either the autonomic nervous system or the amount of light that's coming in. And obviously, if it's low light, we're going to want that to dilate to allow more light to come in to increase the vision in the darkness. And if it is super bright, you're going to want those muscles to constrict. And they're technically always muscle constricting, but we're going to shrink the size of the pupil to allow less light to come in so we don't burn the back of our eyeball called the retina. So the iris is the colored part, which we'll take a look at on some of the models. And then the next structure, obviously light doesn't pass through the iris, passes through the pupil, and then it hits the lens. And the lens is the next structure that is responsible for focusing this light. We want all this light to come in to target a specific spot on the retina, and I will show you what that is. And again, you have the list on the previous page to kind of help you out. So light comes in, hits the cornea, goes through the anterior chamber, passes through the pupil. Pupil's surrounded by the iris, which is the colored part. Hits the lens, and then we enter here, which is called the posterior chamber or the posterior cavity. And this is filled with a fluid called vitreous humor. So we have a front chamber filled with fluid, aqueous humor, the back chambers filled with fluid, vitreous humor. And it's very important for keeping the shape of the eyeball and allowing light to pass through, etc. And as we get towards the back here, you will see this area has multiple layers, right? So the first layer that the light's going to hit is called the retina. And that is this yellowish layer right there called the retina. Kind of, yeah, I'm pointing to it. The layer behind that is a darker layer, which you can kind of see here in red. I'm sorry, it's called the choroid. Choroid. And then the outermost layer, God, I was pointing to it, I labeled it, which you could see over here. It's called the sclera. And the sclera is the white part. So from the outside in, it goes sclera, choroid, retina. From the inside out, it's going to be retina, choroid, sclera. The retina is made of nervous tissue. It's made of rods and cones. Those rods and cones are very, very important. They are called photoreceptors. And those photoreceptors detect light and convert it into an action potential. And then it will send that action potential through the optic nerve. Those messages go back, hit the primary visual cortex of the occipital lobe, um, and allow you to see. So, pretty important. Some other structures that we will talk about with the eye are some muscles. So, you can look up, down, left, right. Your eyeballs can rotate a little bit. So we have some muscles, and I'll show you on on the model because you can see it really good. Um, where these muscles are found, we can see a few of them listed here. The inferior rectus, the superior rectus. We can see the inferior oblique, and we can see that's the only ones we can see on this picture. Um, so the inferior rectus, and I'll show you, is found at the bottom, hence inferior. Superior rectus is at the top. The inferior oblique kind of runs along the side. We will also have a lateral and a medial rectus muscle. When we get to the other picture, I will show you where those are. You have a lacrimal gland and a lacrimal duct, right? You don't have to stress too much about the duct. Obviously, that's going to drain out the gland, but this is where our tears are produced, and they come down and drain through the lacrimal sac, um, and then out. You can see them there. I think there's one model. You can see the lacrimal gland. I'll mention it. This is our best picture. So I'm going to go through here and I'm going to highlight some of the structures that we talked about previously, as well as some of the ones that weren't really listed on there that I wrote in. So light comes in. We can see the cornea, the pupil, the iris, and the lens. Right? This is a little bit more zoomed in. It's very good. It has everything labeled. We can also see the anterior chamber. And now we see that term aqueous humor, right? The chamber's the space, the cavity's the whole thing. You don't have to stress about that. Anterior chamber filled with aqueous humor, right? We can also see a structure called the ciliary body. The ciliary body is located right here. Do a different color for you. Yellow. And you can also see it over here. These are very, very important structures. So you have the ciliary muscles and the processes. In general, we just call it the ciliary body, totally fine. But you can see that there's these ligaments called suspensory ligaments that attach to the lens. So what these muscles can do is they can pull or relax and they can actually change the shape of that lens. That process is called accommodation, and it allows you to focus on objects that are close or objects that are far away by adjusting the lens. I always relate it to a cell phone, right? If you have your camera on and you tap the screen, it goes and it automatically focuses on something. If it's close, you want to zoom in on that. If it's farther, you tap in it, you zoom on it like that. And that's basically what these ciliary bodies are doing all the time. Whenever you look at something near or far, they will adjust the lens to allow you to focus that light. And I will show you where they focus the light. They focus the light right there, towards the back of the retina on a specific spot, part of the macula, we call the fovea centralis. They have a high concentration of cones, very acute vision. Obviously, some of that light will spread out to the side a little bit. Um, but issues with that, if the light focuses too near or too far and scattered light is hitting that, causes blurry vision. Right? We have our posterior chamber, right? They call it the vitreous chamber. You can also call it the posterior chamber, and it contains that vitreous unit. That's in the back. Light passes through, boom, boom, boom. And then our layers that we can see here, retina, choroid, and sclera. And I love this picture. It kind of folds them out so you can see the inner, middle, and outer. The sclera is the white part of your eye. The choroid is a vascular part, so it's supplying blood flow and everything like that to all those other the important structures like the retina. And then the retina again is made of nervous tissue, those rods and those cones, those photoreceptors, very, very important. We have the optic nerve, which I said before is connected to that retina. And as that retina receives light, that optic nerve, cranial nerve number two, which we mentioned before, will send those action potentials back to the brain. And lastly, we have a structure called the optic disc. And the optic disc is where the retina comes in and turns into the optic nerve. There are no photoreceptors there, right? It's kind of, you could just see it's like a little circular area. And we call that the blind spot. Because there's no photoreceptors on there, you don't have any vision. The blind spot's located basically like right there. Um, there's a simple test that you could do to kind of see the blind spot. Um, maybe I'll talk about that later. But that summarizes pretty much all the stuff you got to know on the eyeball. So now I will go through. Here's your list. And I will show you where these structures are on the models. We can see here the pupil. That's the dark part of the eye. You can see the iris, right? That's the colored portion, and you can kind of see it's got like ridges. Those are the muscles. On this view, we're looking at the anterior view. So you can see the superior rectus, the inferior rectus, the lateral rectus, and the medial rectus muscles. We know that. I just know that because this is a tendon here of the superior oblique, and that's always going to be on the medial side. So you just remember the picture or, whatever works for you. We can also see the white part, which is the sclera. Makes sense. This is the same thing, except we can see that lacrimal gland, right? Just different model. You can see those muscles, you can see the iris, you can see the pupil, and you can see the sclera as well. Now, this is a good view because it allows us to see not all, but most of the muscles. We're looking at a lateral view. Here we can see the optic nerve. This is our lateral rectus. The superior rectus is at the top. On here, you can't really see it. You can just see that kind of that piece, but that piggybacks off the picture we just looked at. This is our medial rectus that we can see right here. And then this would be our inferior rectus towards the bottom, right? So real quick, I will redraw that for you. Lateral rectus, medial, inferior, and the superior is up there. And those are our rectus muscles that move your eyeball around. Obviously, you can see the sclera on there as well. Same thing here, right? This actually allows us to see the inferior oblique muscle as well as the inferior rectus. So that oblique kind of just goes to the side. This is the only picture I really have, except for maybe one picture from your real anatomy. Superior rectus, lateral rectus, medial rectus, pupil, iris, sclera, and you're pretty good, right? This is a good model. It allows us to see all the structures that we talked about. Um, and I will go through and show you what they are. Same pattern I went through before. First, light hits the cornea, which would be located right there. Then we have this anterior chamber filled with aqueous humor. We have the opening, which is the pupil. We have the iris, which I'll do in a different color for you. I'll do it in red. That is the iris. We have our ciliary bodies over here, which connect to those ligaments we discussed. You have your lens. Probably not a very good color for that. That's your lens. This is obviously your posterior chamber back here. You can see the three layers. Retina, choroid. That's not the best color. Retina, choroid, and sclera. From the inside out, you can see your fovea centralis. You can see your optic disc and your optic nerve, respectively. So just be familiar with these structures. Obviously, in lecture, we talk more about what they do and how it works. But as far as labeling and recognizing things on the real anatomy, the models, everything I'm going to test you on, this is a pretty good picture. Obviously, we have our cadavers. Everything on here is labeled. We can see the inferior oblique, the inferior rectus. You don't have to worry about ocular motor nerve right now. But be familiar. You can see the sclera. You can see the cornea. Makes sense. Now that you know the basic anatomy, I picked a few slides. And as we progress through with the next, I don't know, 5 or 6 labs, we're going to start to see a little bit of histology. Histology is the study of tissue under a microscope. So I tried to simplify it. The main thing you want to do is recognize the structures. So if I show you a picture of the histology slide, for the most part, and some of them there's a little bit of detail, for the most part, just be able to recognize this is the cornea, right? All these slides are labeled. You could, however you want to go about doing it, but you need to be familiar with them. The next slide is showing us the 3 layers of the eye we discussed. I will show you which ones are which. This is the sclera. I'll label it on here for you. This is the sclera. This is the choroid. And this is the retina. The separation happens here and here. The retina always looks like a zebra. We could see that it's got all those rods and cones in there and different types of cells. The choroid is the darkest layer. It's vascular. We can see actually red blood cells in there in this picture. And obviously that sclera is the white part. So that'd be outside to in, according to that histology picture. This is a picture of the ciliary body or ciliary processes, those little things I talked about that pull on the lens. This is a picture of the iris, right? Those muscles that we'll discuss in a little bit. We already kind of talked about them too. This is a picture of the lens. It's made of crystalline proteins and things like that. clearly very, very important. But this is what the lens looks like under a microscope. And this is a retina. And it has multiple different layers of cells. And talked about it. The light goes in, the action potentials go the other way. Rods and cones are the main point. And you can kind of see the striped nature of this, which is easily recognizable as the retina. This is the conjunctiva. We didn't really talk about it. In lecture, we probably mentioned it. Just recognize it. Probably won't ask you, but recognize it. I would talk about that with a condition called conjunctivitis. Right? Maybe I will ask you conjunctiva. Be familiar. So we have some pathologies, right? There's four that I want you to be familiar with. The first one is conjunctivitis. We just talked about the conjunctiva. Makes sense. Obviously, if you get an infection there. So this picture is conjunctivitis. This picture is glaucoma, right? We can see the opaqueness and whiteness of the eye. Clearly, you can see lots of blood vessels and things like that. This is caused by specific issues with the draining of the fluid in the aqueous or the aqueous fluid in the anterior chamber. We'll discuss it. And we have cataracts, clearly the cloudiness of that lens, build up of protein and such. And then we have something called retinitis pigmentosa, where you can see there's excess pigment formation on the retina, and it makes it very, very difficult to see, and it will clearly obstruct your vision and decrease what the viewing window that you can see out of. That's it for the eye. Know your parts. Obviously, for lecture, we know detail about what it does. Be able to label the histology slides and be able to recognize the four conditions for the pathology. The next thing that we get into is the ear anatomy. It's pretty straightforward. I give you a list of what you need to know, and I will show you in the same process. Instead of a light wave, we're going to be talking about a sound wave, right? Sound waves are basically more or less vibrations of air that can travel from some type of sound source, and they are going to enter in through the outside of the ear. The outermost portion of the ear is called the auricle. Probably won't ask you that, but that's what it is. And then that outer portion of the ear has multiple parts as well, like the lobule and stuff like that. But the key thing is that we have three basic regions of the ear. We have the external ear, the middle ear, and the internal ear, right? And let's see if I can move this out of the way. Accidentally muted myself. That's the picture I'm looking for. You can see that the color-coded regions separate the external ear, which is basically that auricle and the external auditory canal. I'll color code them for you. Right? That's the external ear. And then you hit the tympanic membrane. That's your eardrum. And then from there in, you will notice that you have three little bones. These are called the malleus, incus, and stapes. They're super small. They are part of the middle ear, right? Because they're on the inside. And what they do is as the vibrations come in through the external ear, they vibrate that tympanic membrane, and that tympanic membrane will vibrate the ossicles, as they're called, and then it will vibrate the fluid that goes through the cochlea, which we'll talk about in 2 seconds. And then you have certain hair-like receptors that will detect those vibrations and then will send it in through the cranial nerve called the vestibulocochlear nerve, right? And I'll discuss that a little bit. What color is it? It is peach. I don't have peach. It's all good. We'll use purple instead. So as we get inside, you have the inner ear structure, which is made of these big rings and this big snail-like looking guy, right? The rings and the snail. The snail is called the cochlea. The cochlea is a highly specialized organ designed for hearing. And we'll take a look at that in a few minutes. We'll cut it open and we'll go inside and we'll see what we can see. The semicircular canals are for equilibrium and balance, as is, it's not labeled on here. This middle region is called the vestibule, which we'll be able to see on a different picture. So we have three basic regions. We have the external ear. We have mainly made of the external auditory canal and the tympanic membrane, as well as the auricle on the outside. You have the middle ear, which is made up of the three bones, malleus, incus, and stapes, also known as the hammer, anvil, and stirrup. Care if we use that terminology, but those are the words we have here. And we have the inner ear, and inner ear infections and things like that, which is made up of a few structures called the semicircular canal, the cochlea, and the vestibule, right? And that vestibule is that guy right there. And because we can see it on this picture, and I already mentioned it, the vestibulocochlear nerve, cranial nerve number 8, has two different branches. One of these branches runs to the cochlea, and it is called the cochlear branch. That is for hearing. The other branch is more for equilibrium, balance. We'll mention vertigo. Hopefully not, but you know. And that is the vestibular branch. That runs from the vestibule and is more involved in balance, things like that. Other than that, we have one more structure, and that is the auditory tube. So as these vibrations come in, right? Vibrate, vibrate, vibrate. Obviously, these sound waves are coming in. They vibrate the bones. There's fluid inside this cochlea. That fluid vibrates. Those receptors detect it. We have to have an area for these vibrations to leave. So those vibrations will exit out of the auditory tube. And that's where it is. Goes to the nasopharynx, pharynx, back of the throat, etc. On this picture, we can see a couple things. Right, we can see the auditory tube, the tympanic membrane. That whole middle and inner ear structure is removed on this model. And then this would be our external auditory canal. So we can see a couple of those basic structures, more or less the tubes and that tympanic membrane. This is showing us an, let's call it zoomed in view of kind of that transition between the external to middle ear. And we can see our basic structures we talked about, the tympanic membrane, the external auditory canal. We can see the malleus, incus, and stapes. A structure we probably have talked about already is called the oval window. And the oval window is where that stapes, you can see it has an oval-like shape. That is where it attaches to this inner ear structure, right? I think I have a model. I will show it to you. And clearly the auditory tube as well. This is just a picture of the three bones. So I will highlight them. This is the stapes. This is the incus. And from this view, you can only kind of see it. That's the malleus. Here we can see the malleus a little bit better. That's our hammer. Forget what color I used. I think it was orange. That's the incus. And that is the stapes. Again, this model's about yay big. Clearly, these bones are very, very, very small. So they just increase the size to show you the detail. Here, we can see the cochlea. Lovely. You can see that there's a cochlear duct in there. For lab, you don't have to stress about that. You can see that positioning of that oval window we discussed where that stapes attaches. You can see the round window here. I didn't mention it previously, but it's important for transmission of those vibrations as well. You can see your semicircular canals. You don't have to know the specific ones. Just know that they are semicircular canals. Obviously, they're filled with fluid as well, endolymph and stuff like that. And lastly, you can see the vestibule. We consider this entire area the vestibule, more or less. It is made of two structures called the utricle and the saccule. Probably talk about that in lecture, not so much in lab. So be familiar with the vestibule, and I'll show you where it is on this model. Here we can see the cochlea. The vestibule is in this region here. And these are our semicircular canals. That's the big picture, right? You can see a little bit of the oval window down here. Maybe I'd ask you off this picture. If we have a better view, I'll show you that. There's our better view. That's the oval window. That's the cochlea. Those are the semicircular canals. Makes sense. Here, same thing. Still looking at the inner ear. Let's label what we know. This is our cochlea. These are the semicircular canals, right? Here is our vestibule. And the new thing that we're adding here are the two branches. That is the auditory, or I'm sorry, the vestibulocochlear nerve, aka auditory nerve. But here we can see that this branch runs to the cochlea. That's the cochlear branch. This branch runs to the vestibule. That's the vestibular branch. Combined, they're the auditory nerve or vestibulocochlear nerve, cranial nerve number 8. And this is a pretty good picture to see that differentiation of those two branches. We can also see that here. Cochlear branch. I think it was the other color. Cochlear branch. Vestibular branch. Obviously, we can also see the vestibule, semicircular canals, and the backside of that cochlea. This picture, we open up the cochlea. And I'm going to show you why this is important. So nothing new per se on this picture, but this, once we open it up, one of those tubes, if you will, of the snail, we're going to zoom in on, right? Because I want to show you what the inside of that structure looks like. And that's what we can see here. So there's a couple things we got to know. Move that out of the way. That is the inside. We took one of those snail tubes, cut it, and looked inside of it. This is what it would look like, right? So there's a couple important things here. Number one, we have 3 spaces. I'm going to do it in a different color. We have a space at the top we have a space at the bottom, and we have a space in the middle. They have names. You need to know all three. The top one is called the scala vestibuli. The bottom one is called the scala tympani. And the middle one is called the scala media or the cochlear duct. So we learned the basic structures from the outside in, external, middle, and internal ear. And then we took that cochlea, part of the inner ear, and then we cut it open, and then we looked at the inside. The inside of that snail, right? One of those tubes has three little tubes. They're filled with fluid. It tells you what type of fluid. For lab, you don't have to know. Paralymph, endolymph, etc. Um, and the important chamber, I mean, it's all important, is this middle guy. And that is because that is where we find the organ of Corti. And that is this guy. This is what allows us to hear. This is what vibrates when that fluid vibrates. And the specific thing that's vibrating is called the tectorial membrane. And I'll show that here. Right? This thing vibrates and it's tapping on these little hair cells. Worst color ever. It's tapping on those little hair cells. Then we depolarize. We send messages in through the auditory nerve. There is two more things we must know. And I will do them in a darker color so it is not confusing. The first one is called the vestibular membrane. And that is the membrane that separates the scala vestibuli from the cochlear duct. And the second one is called the basilar membrane. And that's what separates the cochlear duct from the scala tympani. And that's the gist. Anatomy-wise, you're done, right? These are our chambers, scala vestibuli, scala tympani. This is the organ of Corti. That is your tectorial membrane. This is the cochlear duct or scala media. And then respectively, we could see the two membranes we discussed, vestibular and basilar membrane. Right? We have some histology, not a lot, a couple histology slides that I want you to be familiar with. The first one is a picture of the inside of the semicircular canal. The next one is a structure called the crista ampullaris. We, at this point, probably have already talked about it in lecture. This is just showing you a histology slide of that. This is a picture of the cochlea, right? Mainly we're talking about the specific receptors and things. On here, it's very similar to the picture you just looked at, right? You can see that middle chamber, the top chamber, what you want to call it, cochlear duct, and the bottom chamber. And here, this is where your organ of Corti will be. And again, just recognize this as the cochlea. This shows some detail. I don't really have a good histology slide of this. That's why I use this. Of those hair cells and that tectorial membrane, cochlear duct, and things like that. But this is the organ of Corti. Now, we're almost done. There's a couple tests I want to discuss, and then there's a little chart that you're going to fill in afterwards. I do want you to be familiar with the images, right? So I picked some specific tests. Some of them have pictures, right? The astigmatism, the visual acuity, the blind spot, I will show you what the test looks like. I'll draw it for you in 2 seconds. Dominant eye, I'll show you how to do. Lens focusing action, kind of discuss that. As objects move from close to far, we have a process called accommodation. And then the light reflex is the changing of that pupil that we discussed, right? AKA the iris. First one, oh, let me show you the blind spot test. Basically, you get a piece of paper, and you can do this, right? You just get a piece of paper, whatever, and draw a circle and an X. And then you close one eye and you stare at the circle and you have to stare at the circle. You slowly move this thing back and forth, and you'll get to a spot where that X disappears. When that X, the light reflecting off it, hits your blind spot, won't be able to see it because there are no receptors on the optic disc. Uh, so that's what it looks like, that little circle and the X, and you just stare at the circle, and then the X will disappear like it just did. Even on my computer, it works. Some other tests. This is the astigmatism test. I'm not going to go through crazy detail how they do this, but you stare at it, and some of those lines will kind of fade. They'll turn gray or disappear, and you can't really see it, but there's numbers on there. This is a blurry picture. You don't have an astigmatism, or maybe you do. And that allows them to diagnose certain aspects of it. This is visual acuity. This is how you do your vision. You have 20/20, 20/15, 20/30, 20/40, whatever. And what you would have to do is be able to read these letters at a distance of 20 feet. And the smaller you can go, the better visual acuity you have, right? It's a vision test. This is showing you what I talked about, and I thought about bringing it up in the PowerPoint, but like earlier, but I figured we'd get to it, so it doesn't matter. This is your pupil, right? So the big picture here is we have two types of muscles, circular and radial muscles. During parasympathetic stimulation, right, the circular muscles close the diameter of the pupil. During sympathetic stimulation, the radial muscles pull it out, dilating the pupil, right? We also talked about this reflex in response to light. Should be familiar with that. Super bright, right? Dilate. Again, not the best color, but it's all good. Dim light, we're going to die. Sorry, super bright light, we're going to constrict. Super bright light, circular muscles. Dim light, dilation, more light coming in, radial muscles. Makes sense. We don't have any, obviously you can't do it because you're online, any taste tests or things like that. There are tests that exist, but what I do want you to be familiar with, when we get into digestion, I'll show you pictures of like the taste pores and what makes up the tongue and stuff like that. But for now, you should know your 5 tastes: bitter, sweet, salty, umami, sour, right? And lastly, pretty straightforward, not the worst lab. Um, I want you to be able to fill this out, right? And this is a chart. You don't have to go super crazy. You will find this stuff in your book. But I really want you to put a heavy focus on the receptors. Not necessarily that I'm going to ask you a tremendous amount of it in lab, um, but because we move so fast, it's very important that you kind of pair this together with lecture. So this is just practice. Really, we talked a lot about the vision and the hearing. The equilibrium and olfaction is definitely more lecture. The cutaneous, we've talked about that with sensory pathways and things like that. So take a look at it. Focus primarily on vision and hearing, and some of those tests. And that about wraps it up. So, hardest part, anatomy of the eye, anatomy of the ear, histology, couple different conditions, um, and a few tests. Pretty straightforward. I will see you in the next one.